So good afternoon everybody. So today I will be starting my class. So I will be talking about an important aspect of in your syllabus that's present in unit 2, and that is data, systematic data and types of systematic data. So this term is very confusing. Systematic data is data which is obtained in systematics or taxonomy. Taxonomy is the science of classifying animals. You have to name them, you have to on the basis of characteristics, you have to put them under certain positions in the taxonomic hierarchy. Then you have to take these animals and then you have to assign characteristics, you have to collect specimens of them, and then this is taxonomy. And in systematics you do all these things. On top of that, what you study is that you study the evolutionary relationship between these organisms, you study the behavior, adaptation and all these things. So definitely there is a lot of data involved in this particular aspect. And what are these data? This is what we are going to talk about right now. So taxonomic data are taxonomic characters. Basically they are characters. They are taxonomic characters. So Ernst Mayr was one of the champions who classified taxonomic characters. And so I will in the book Mayr and Ashlock 1991, it's perfectly written over there what are taxonomic characters and problems. I would recommend you people to study from that book. And from that book, at least this chapter, I'll mix up couple of books because all these things you don't have a single textbook available. And the university has recommended a book by Quirk, Principles and Techniques of Contemporary Taxonomy. They have been recommended. I looked at the price in Amazon, it is 10,000 rupees. So we cannot buy those books and we cannot use those books, but we have to gather information from other aspects. So to start, let me start sharing my screen. So as I mentioned over here, I've written over here, I've uploaded this particular scan in the Google Classroom today itself. And this is Mayr and Ashlock 1993. I've underlined and I've highlighted regions which is important for you to study. So taxonomic characters is also taxonomic data, or you can call them as, it's also synonymously known as systematic data. So what are taxonomic characters? So taxonomic characters provide the evidence from which relationships between taxa is inferred. So what is a taxa? Taxa is phylum, subphylum, class, order, genus, species. These are known as taxa. So you have a cat and a dog. Both of them belong to the order Carnivora because they eat meat. So on the basis of this carnivore, and then you go to class Mammalia. In Mammalia, what you have cows and you have dogs and cats. Cows are vegetarian, they are herbivorous, and they are these are carnivorous. So they belong to different subclasses, different orders. Sorry, they belong to different orders. They belong to order Artiodactyla because their hoofs, they are bifurcated, and they belong to Carnivora because they eat meat. So on the basis of this, this is a taxonomic character. Yeah, a relationship is inferred. Now cats and dogs have been put under order Carnivora because both of them eat meat and they have very firm carnivorous teeth, canine teeth, and on this basis of this character they have been placed in the same category. And Lophodontata is not placed. The rabbits and the rodentia, they are not been placed. The rabbits have got a different type of, they are rodent like teeth. So the chisels are higher. The canines are not very well developed. Canines are the ones with on the side, the pointed sharp teeth, vampire teeth. Those are your canines. So taxonomic characters, they provide the evidence from which relationship between taxa is inferred. So according to Mayer, the definition is a taxonomic character is any attribute by which a member of a taxon differs or may differ from a member of another taxon. So how does order Rodentia, that is of rats and rabbits, differ? It's a taxon. It differs from another taxon, which is order Carnivora. That is their teeth and their diet. They differ. Any difference between two individuals is a character, but all characters are not useful for taxonomic purposes. So knowing where useful characters are to be found and establishing their specific value is the most important skill of a systematist or a taxonomist. It requires good theoretical knowledge as well as experience. There are thousands of characters. They have legs. Cats have legs, cows have legs, we have legs, insects have legs. Well, is it a useful character? Well, it is doubtful. What type of legs? That may be useful. So this, I know what type of legs because I have some experience. I know that there's some legs, insect legs are thoracic and it's different, and these are the legs which we have or cows have. They have got, their origin is the same, so they are different. It is not thoracic legs. It is a limb, that those are limb bones, so the development is different, everything is different. We have got former knowledge, therefore you require experience. Okay, now inside what do you mean by difference? Must be between two different species. In the same species, for example, say maybe you are talking about a population of dogs. Some dogs are white, some dogs are brown, some dogs are male, some dogs are female. Well, these are not sex is not a taxonomic character. Coat color in dogs, in a particular population of dogs, it is not a taxonomic character. For it to be a taxonomic character, it has to be a different species, or different, not a species, different taxa. For example, it can be a dog and a wolf. Wolf are different species, but their generic name is the same. So they must be in a different taxa. So this is the basic essence of taxonomic characters. Now there is another term has come up, that is known as signifiers. What is the difference between that of characters, and what is the problem if you use it is a character? So a character is a property, attribute or feature that distinguishes one thing, individual or group from one another. Some character which distinguishes one group from another group. So black feathers are a character of Australian swans, and white feathers are a character of European swans. This is a taxonomic character. Now in 1950s, computers started coming up, and to be handled by the computer, all characteristic must be, all the characters, these characters, they must be assembled into a table known as data matrix. A table was made. These are the you had the characters 1, 2, 3, 4 characters were written. Maybe here you give the name of the species, species 1, species 2, species 3, species 4, species 5. Oh, whether this character is present in species 1, yes, you give a plus. Oh, whether this character is present in species 2, no, it is -1. Whether this particular character is present, no, minus. This is plus. Like this, a table like this is a data matrix which you construct. So this type of a data was very important when you worked with a character. So the columns for the table are for taxa. So these are columns. you can give these columns as taxa, and each row contains the numerical code for the characters. These rows have got the numerical codes for the character: plus, minus, -1, -2, -3, -4. Then what was the character? Problem was what to call the rows, because you can't give the name character over here. So to differentiate this, to solve these problems, they transferred the term character from the difference between taxa. For example, if we used black feather versus white feather, so where would you keep black feather and where would you keep white feather, and where would you give the species name? If you make a column and row classification, you could not give. So for this, they started using the term feather color. For example, they started to refer to the structure that varied. So now you gave the name of a character as feather color, and now you can give it black. This is species 1, species 2, species 3, species 4. You can give it black, you can give it white. This another species, you can give it black. So like this you started giving the names, and this started to be called as the character state. So instead, black feather, white feather, they are characters, but when you call them as feather color, then that is the character state. The feather color can be either black or it can be white. For example, Drosophila's eyes, it can be red or it can be white. Red is a character, white is a character, but eye color is a character state. I hope I am clear in this part. So this proposal had the unfortunate result that the term character is now used in taxonomy in two very different senses. In computer terms, character state, you started to give character state, while in the classical terms you started giving black feather or white feather. So some people started talking about putting black feather, white feather. Some people started talking about eye color plus, eye color minus, or eye color black, eye color white. So they started talking about character state. So this was a problem. So to solve this problem, Ashlock in 1985 proposed restoring the term character to its traditional meaning, and that character is either black character or white character, and introduced a new term known as signifier for a feature that varies from one organism to another. The feather then would be the signifier or sign bearer, and the feather white and the feather black would be the character. So the signifier became the character state. The character state. So the feather became the signifier. The character state became the signifier or sign bearer, and white feather and black feather, they became the character. Thus, character particular in regard to numerical methods refers to a variable feature, and character state is defined as an attribute by which a member of a taxon differs from a member of another taxon. So now we go to what are the different types of characters. So let us look at this table. Table 7.5, overview of taxonomic characters. Or first one is the morphological characters, or some people have written it as morphological data. They can be, morphology is external features of an organism. how, whether it is tall, dark, whether it is circular, whether it's square, how you look, that is morphology from outside. Then there can be special structures, for example the genitalia, that is an important character on the basis of which you can classify an organism, or you can classify an organism whether it belongs to this class or whether it belongs to that other class. Then internal morphology, this is also another word used is anatomy, internal morphology. It can be embryology or the development. So one organism develops different from another organism. So on the basis of this development differences, close species, you can differentiate close species into different species. Then you have karyology and other cytological differences, looking at the chromosomes. Apart from that, you have physiological characters. They can be metabolic factors, they can be body secretions, they can be genic sterility factors. I will come to these factors. Genic sterility factors means, so suppose you cross a male donkey with a female horse, you get a mule, but this mule is sterile. So horse and donkey are of different, they are different species, they belong to different species. So this is known as genic sterility factors. You have molecular characteristics, you have immunological antibody data. So close, then you have by electrophoresis you can differentiate the DNA. Then you have amino acid sequence of proteins. Another important technique is DNA hybridization. Then you have DNA and RNA sequences. Then you have restriction endonuclease analysis. Then you have other molecular differences which we talk. Then you have the behavioral characters, most important being courtship or mating pattern. We will talk about this very soon. Then you have ecological characters. Ecological characters can be habitats and host, food, seasonal variations, parasites, several host reactions we will talk. Then we have the geographical characters also. So let us start now with morphological characters. What are morphological characters? Features of external morphology vary according to kind of animals. They range from superficial features such as plumage and pelage. Plumage is the feathers of a bird is known as its plumage. So for example, all parrots, they have got green body and red beak. So plumage, that is a characteristics. Pelage, the beak, red, that is parrot. You can classify parrots from crows on the basis of plumage. Crows are black, parrots are green feathers. So this is morphological characteristics. In some of them, internal anatomy also may provide many taxonomic characters. I have marked it in black. You can read this part. Only in the last two parts you have to read the whole thing. In the last two characters, geographical and ecological, I would recommend you to read the whole thing. Internal anatomy provides many taxonomic characters in practically all higher animals. So in the preparation of mammal skins, the skull with teeth is routinely preserved and used in classification. So on the basis of the skull and the teeth, you can say these are hominids. Monkeys and humans, they have got similar skull, and so they are hominids. While reptiles, amphibians and fish are normally preserved in alcohol and are always available for dissection. Then you have the fossils. Fossils, they consist almost entirely of preserved hard parts, because you will not find soft part in fossils. So on the basis of the fossils you can also classify, and they consist of teeth. From the basis of teeth you can classify organisms. Okay, this type of teeth, so they are order Carnivora. This type of teeth, they are herbivores. You can differentiate these species. So hard parts and the work of animals. In invertebrates, the shells and exoskeletons important. Among protozoans, test shells, thecal plates, cysts and other hard parts are vital in the classification of foraminiferans, radiolarians, testaceous rhizopods, flagellates and other organisms. Then the calcite crystals in the skeletal of echinoderms. Echinoderms are starfishes. You have calcium crystals. They have a specific shape. On the basis of that shape, you can classify them into different families and orders. Some have got calcium, like you have Asterozoa. Then in holothurids or sea cucumbers, you don't have them. So on the basis of this, you can classify them separately, these starfishes. Then dinosaurs are based on fossil tracks. Dinosaurs of classified. Then gall insects. Gall insects are insects which form little tumor-like structure on certain plants. That is known as gall. So these in gall insects, sometimes they yield a good clue to relationship as do the insects themselves. They form the mines. The formation of a mines is an important taxonomic characters in mining insects. Mining means they make holes in the leaves. So the pattern of the hole, by seeing the pattern of the hole, the two insects can be said to be close. If both the insects make same type of hole, then they belong to the same group. If they make different types of hole, they can be of different group. For example, this figure you can practice. The holes are different in different. In this leaf you have a different hole. In this leaf you have a different types of holes. In this leaf you have a different type of holes. So on the basis of this, they can be differentiated. So in taxonomy, more than how similar it is, how different it is, that is more important. Next is coloration. Color pattern is the most recognized and it is the most convenient character. Every species of birds, as I mentioned, can be recognized by its color. Then same with for reef fishes and for butterflies, by its color. Then the genital structure. In insect, this is very important. For example, species like Drosophila melanogaster and Drosophila ananasi, they have got a genital plate in the genital region. If you boil out all the part, this genital hard part remains. And then comparing the two structures of the genital part, the small fruit flies or Drosophila which you see hovering over your fruits, you can distinguish these flies on the basis of their genital structures. Then different sperms, the structure of spermatozoa of different insects, they have different morphologies. These are the different morphologies which are present. On the basis of that, you can differentiate two species or two groups. Among other characteristics, behavior is used. For example, courtship behavior is used. Then larval stages and embryology. So the developmental various immature or larval stages of embryos and sometimes even eggs may provide taxonomic information. The various sibling species of Anopheles maculi penis complex malaria mosquitoes were discovered because of differences in egg structure. The eggs are of different groups of Anopheles. They are different. Sibling species, they look alike, but they don't mate among each other. Or even if they mate, they have got different sterile hybrids, like Drosophila melanogaster and simulans. I will show a picture later one day that they look absolutely the same, but they are basically different species. So in Anopheles, different species of Anopheles, they can be distinguished by the nature of the eggs. The eggs look different, but the flies are the same. So they have been kept in different groups. In groups with complete metamorphosis, entirely different sets of characters evolve in adults and larvae, and conclusions drawn from the character of one stage from a very useful check on conclusions drawn from those of another. So you look at a development of two groups. One group has got a development from larva to adult, and another group also has got a development from larva to adult. Now you look at the intermediate stages. Are the intermediate stages the same, or they look the same? If they don't look the same, then they are different. Those groups, you can keep them in different groups. Then we have genetic characteristics. Obviously all changes which take place, they take place at the level of genes, DNA and RNA. Sterility, like I talked about the example of a mule which is sterile, formed for result of cross between a donkey and a horse. The mule is sterile. Then chromosomes. Basic studies of chromosomes, they come from polytene chromosomes, or the salivary gland polytene chromosomes. In Drosophila, two species, Drosophila simulans and Drosophila melanogaster, they can be distinguished very clearly with by polytene studies. So they are thick chromosomes like this. From a center you have different chromosomal arms, thick arms coming. They can be seen under microscope very nicely. 5 arms are present in Drosophila. So on the basis of the banding pattern and the nature, size and the number of these arms on polytene chromosomes, you can these are the chromosomes present in the salivary gland of the larva. You can actually say that they belong to the same species or not. Then you have polyploidy and all these examples are given over here. You have to read this chapter completely, and the parts underlined are the most important parts. So I will stop my class today here. Next day I will start with physiological characters. What are physiological characters? Now to see if anybody has got any questions. If you have questions, please raise your hands. I know this is a bit boring, but this is what you have to study. In my section is a very boring section. You will enjoy the cladograms. Of course, that is an interesting part. I'll teach you how to construct cladogram, but the rest are all are rules which you must know on how to do. But it is very important, not only in zoology but also in botany, microbiology, whatever. You need to know all these things. So if you people don't have any questions, if you have questions, please raise your hands and please ask. Shreya Singh, do you have any questions? Are you able to hear here? Hello. Okay, you don't have questions. ठीक है, आवाज मिल रहा है. Okay, fine, very good. Fine, then nobody has questions. If you have questions, then you can raise. There is an option called raise hand, or you can even write chat, type a chat. Okay, so there are no questions. Then I end my class over here. From next day I will complete the taxonomic data, the systematic data. I have uploaded this note in the Google Classroom along with another note also present. There are several things which I uploaded. Keep on reading those notes. Don't wait for the classes. Anything can happen. Classes will be cancelled. Okay. Wishing you all the best of luck. Stay safe.